For this project, we'll be breaking down and understanding the complex process of the electron transport chain. Before we begin, we need to orientate ourselves on where exactly this takes place in our bodies. The mitochondria, also known as the powerhouse of the cell, can be broken down into different parts. First, we have the outer mitochondrial membrane, which is like a skin around the organelle that is holding the mitochondria together and faces the cytoplasm of the cell. Inside we have the matrix, which is a fluid that contains smaller organic molecules and is also where the citric acid cycle occurs. The colored yarn is the cristae, which are the folds inside the mitochondria, and the inner mitochondrial membrane is also located right here. The space between the inner mitochondrial membrane and the outer mitochondrial membrane is known as the intermembrane space. Now that we have a basic understanding of the mitochondria, we are going to zoom in on the inner mitochondrial membrane, which is where the electron transport chain is located. Now that we are zoomed in on the inner mitochondrial membrane, let's take a look at the overall picture of the electron transport chain. There are 4 protein complexes and ATP synthase. Remember, we are on the cristae, so above us is the inner membrane space, and below us is the matrix. The number 1 rule of the electron transport chain is that we're moving from a high reducing potential to a low reducing potential, meaning that the chain cannot work backwards. We are going to start with 2 coenzymes, NADH and FADH2, which were given to us from the citric acid cycle. NADH is going to donate 2 electrons to this first complex, also known as flavin mononucleotide, and then we are left with this NAD+. So you need to remember that every NADH releases 2 electrons and 1 proton. FADH2 is oxidized as well and becomes FAD+. So remember, oxidation is losing electrons. Now the electrons are going to hop onto something called ubiquinone, and ubiquinone can move freely within the membrane and transports these electrons to each complex. We also have cytochrome, which does the same thing, but it's a little bit too complex, and there's many different ones and different functions that they do, so we're not going to go into depth about that. But these electrons are moving down the chain, and each electron is going to start pumping out these hydrogen into the inner membrane space. 2 hydrogen to be exact. The pumping of these hydrogen through a selectively permeable membrane is also known as chemiosmosis. When we get to the 4th complex, the opposite of oxidation occurs, which is reduction, and reduction is gaining electrons. Now we have 2 electrons, hydrogen, and oxygen. And because of reduction, we get water from that. And this water just dissolves into the matrix. All of these hydrogen are forming an acidic gradient, and they need their energy to go somewhere. So this brings us to our last step, which is ATP synthase. ATP synthase spins, and inside of it has a DP and different phosphate groups, as well as all of these hydrogen. And then the ATP synthase is going to spin, mix all these, and from it we are going to get ATP. And we'll get 36 to 38 ATP. This completes the oxidative phosphorylation electron transport chain. Thank you for watching.